This video is going to cover the DNA mutations out there. And let's go ahead and think at the cellular level for a moment. We have a cell, and in that cell is a nucleus, and the nucleus is our DNA. Our DNA is going to be transcribed into RNA, and that RNA is then going to leave the nucleus of the cell. It's going to find its way to an organelle called the ribosome, and at the ribosome, it's going to be translated into proteins. So right here, we're looking at a strand of RNA. How do I know that this is RNA? Well, DNA has 4 bases. It has an A, a G, a C, and a T. Adenine, guanine, cytosine, and thymine. Now, looking at this, we have a G, a C, a U, and an A. There's no T in here. In RNA, the T that you find in DNA, the thymine, is going to be replaced with a uracil, so the U. So RNA is going to have guanine, going to have cytosine, going to have uracil, and going to have adenine. So that's how I know that this is an RNA strand. There are uracil bases in here, thus it's an RNA strand. So we're looking at the ribosome part now. If our DNA tells us to make this RNA strand is going to use the genetic code in forms of triplets or codons to form our proteins. So this triplet, GCU, forms an alanine. This triplet, AAG, forms a lysine. This CCU forms a leucine. GAG forms a glutamate, and UAU forms a tyrosine. How do I know this? I encourage you, if you're unsure of what each of these sequences is, which most people are, I'd recommend just Googling it. Type in the genetic code triplets, what sequence forms what proteins. You can Google whatever you want. It'll pull up a nice little compact box, and it'll give you what each of these triplet codons are going to translate into for proteins. So here's our normal sequence. We're looking at GCU, AAG, CUU, GAG, UAU. It's going to create our nice protein sequence of this order. And let's say this protein sequence is going to go on and create an enzyme in some random cell of the body. But we need that random enzyme to function. We need our genetic code to create this exact sequence so we can create a nice protein, and that protein can then go on and serve some function in the body. Now let's look at a silent mutation. In a silent mutation, we're not going to be changing the protein sequence, but we are going to be changing the base sequence. So what do I mean by that? Well, we have a GCU, which is still going to create our alanine. We have an AAG, which creates our lysine. We have a CUU, which creates our leucine. And we have a GAA. Look, we have a GA, and now we change a base in the sequence. We change from a guanine to an adenine. And this is still going to be a tyrosine. No difference. However, we need to take a look at this part. We changed one of the bases. We took a GAG and turned it into a GAA. Remember, there are mutations that happen all the time. Our body has ways that we can fix it, but a silent mutation, do we need to fix a silent mutation? Of course, it's a mutation. That's bad. However, what's it going to do to our protein sequence? We have an alanine, a lysine, a leucine. The genetic code is awesome. It has a little bit of wiggle room where a GAA is still going to create a glutamate protein. So we are going to see a alanine, lysine, leucine, and still a glutamate. Even though we changed a base around, this is called the wiggle rule. We have a little bit of wiggle room where a GAG and a GAA are both going to create the same amino acid here. So our amino acid, alanine, lysine, leucine, glutamate, and tyrosine are still going to go on to make that same protein for that random cell in the body. It's not going to be affected. It's still a mutation. It needs to be fixed, but it's not going to affect the outcome later on down the road. So now what happens if we see something that does affect the outcome? Here we have a missense mutation. We still have our GCU, which creates our alanine, our AAG, which creates our lysine. We have a CAU in this case, which is going to have a different sequence than our CUU. We're going to look at our original sequence. So let's actually get rid of our silent mutation there. Let's compare everything to our normal mutation. So we change our order around. We have a C still. We have our cytosine. This case, we have a uracil that's replaced with an adenine. So what is that going to do to our sequence? Unfortunately, it's going to change our amino acid that gets translated. So here we had a leucine, and now our CAU is going to be a and now our CAU is going to be a histidine. So we have a completely different amino acid that is getting put into our protein chain. Our GAA is going to be different as well. We have a silent mutation there, but remember I said that the GAA sequence is still going to create a glutamate. So we have a missense mutation here where we actually change up the amino acid that is going to be inserted into our protein. We have a glutamate, which is a silent mutation, which we just saw earlier. However, it's still going to throw in the same amino acid into our protein chain, and here we're still going to have our tyrosine. So in this case, our protein is different. We have a different amino acid component to our protein. It's going to go to our cell in the body, the random cell in the body, and it's going to have an altered function perhaps because it has a different amino acid. It's not the same protein. It has a slight variation to it. Whether it makes it a better protein, a worse protein, more likely a worse protein, but it's going to change it, alter it. So that is a missense mutation. When you change a base in the sequence and you're going to get a different amino acid. So let's go ahead and look at a different mutation now. We have a nonsense mutation. Here we're going to have a GCU, so we're going to translate that into an amino acid of alanine. An AAG is still going to be our lysine, no change there. CUU is still our leucine. GAG is our glycine, or glutamate, my apologies. Now what about this last one? We have a UAA instead of a UAU. What is a UAA going to translate into? That's not good. We have a stop codon. So we're going to keep adding these amino acids to our chain until we reach a stop codon. And UAA happens to be one of those. So let's go ahead and pretend that since we don't have a stop codon up here, we're going to have a continued sequence. We'll have more until we reach a stop codon, in which case we'll stop translating these amino acids onto our protein chain, and thus we'll stop and finish our protein. Proteins are made up of lots more than 5 amino acids. But here we prematurely add a stop codon. We're supposed to continue. However, in our nonsense mutation, we prematurely add a stop codon to the sequence. We have a mutation in our sequence, a uracil changed to an adenine, and that accidentally truncates our protein. We stop adding amino acids on beyond that. So we have an alanine, a lysine, a leucine, a glutamate, and stop. We are not going to continue on with making that protein. That is a nonsense mutation. These are bad because they're going to truncate that protein. They're going to chop whatever else is beyond this off. So let's say we're only 5 amino acids into building our protein. And we stop. That protein is not going to function right. So it's going to go off to that random cell in the body and completely be out of whack because it does not have the correct amino acid sequence. We chop off all the stuff that it needs for that protein to work correctly in the body. A nonsense mutation is when one of the bases changes and is going to create a stop codon. Finally, let's look at a frameshift. A frameshift mutation is going to be one of the most complex to kind of conceptually think about. So let's look. We have our GCU, which is going to create our amino acid alanine. And AAG is still going to create our lysine. And this is where the difference begins. Notice here that we didn't swap a C for a U, and a U stays the same, and a G and a U. What we did is we're going to take out this C. This C gets deleted. We're going to have a deletion, or we could insert an extra amino acid into the sequence. But here I have kind of represented where we delete one of our original sequence, where we delete the C and just shift everything beyond that into its place. So UUG becomes a codon when CUU should have been the codon. Since we took out that C, now our new codon reading sequence is going to be UUG. So here's our UUG, and then etc., etc. Our AGU is then going to shift over into a codon sequence, and then whatever was beyond that is going to be read. So our AGU, AGU, AU, this should be a U, my apologies. AU, and then whatever else we found beyond that is going to be left off. So what is this going to create? Well, a UUG is going to create a leucine. A UUG is going to create a leucine, while a CCU was also a leucine. So here, notice how this frameshift mutation, even though we deleted this C and shifted everything over, we're still going to get the same amino acid. This is that wiggle room. Each amino acid has a couple different variations on how to create it. And a CUU happened to create a leucine, and so does a UUG. However, we're not going to be as fortunate in the next case, where we have up here in our normal sequence a glutamate. However, our AGU is going to create a completely different amino acid, and then etc., etc. So we have a tyrosine created by a UAU, our AU, and then whatever else was going on after it over here is going to be shifted and in its place. So it'll be like AUC or AUG perhaps, whatever it is. We're going to get completely different amino acids. And this is going to screw up everything beyond our frameshift mutation. Notice how everything before our frameshift mutation stayed the same. We have the same amino acids. However, after our frameshift mutation, we took out that C, we're going to have a completely different sequence beyond it. And that's going to really affect our protein function. So when it goes to that random cell in the body, it's going to have an altered function. So that's some of the basic mutations of DNA. If you have any questions, be sure to ask. Otherwise, we appreciate any feedback you may have. Let us know how we did. Thanks.